Okay, last talk, Jolene in Bradford.
All right.
Go ahead.
All right, thank you. And I'd like to thank you, Andrea, for allowing us to bring this conference to a close. And congratulations to Paul and Andrea for such a great organization of the course. And I'd also like to call out my colleague, Paula, for all of the work that she did to support this. And thank you to all of you for staying for this last little bit. I know it's been a long and inspiring course. I have to say that I'm humbled to be a part of the flow cytometry community and support all the kinds of exciting research that I've seen here today. And at the same time, I'm quite grateful to be a part of the development of many of the tools that you all use. So I'll talk about a few things here. We can kind of chill through this, so if you have a question, wave at me, and if not, I'll just keep on going. And one of the things that I like about groups like this is you have a lot of ideas of how to do things in new ways, and I'd like to hear about what those ideas are or what your problems are that you're trying to solve so that we can work on those same problems together. So almost every presentation here had some sort of live dead cell discrimination, and I'm really happy to hear that. You know, cells exist on this continuum, and in flow cytometry we basically call dead cell identification through the plasma membrane integrity. So this is one of the seminal papers. It's a little bit old, but it's from Steve Perfetto and the Mario Roeder lab. So they did a lot of studies of looking at their PBMCs, and they gated on their scatter parameters for the identification of dead cells. And after a while, they started questioning these results, and so they did a little study, and what they did was gate the way they normally did on scatter to identify their dead cells and their live cells. And then they put in an actual dead cell stain to identify the cells. And when you get over here to the end, you'll see there's quite a different result if the dead cells are excluded. And so really, scatter alone cannot identify your dead cells. So here's some results that were generated in my lab, and it's older blood from a human with CD4 and CD8, and gated on a lymphocyte population. And you'll notice this co-positive population here. But when you eliminate the dead cells from that, you'll see that that population disappears. So you know, antibodies can non-specifically bind to dead cells. So it is really critical to your results to exclude the dead cells from your analysis. Again, looking at membrane integrity, there's really a couple of ways to do this. Looking at the impermeant nucleic acid dyes. So a healthy membrane will exclude the dyes, and a membrane that is compromised will allow the dye in, and it will label the nucleus and become very brightly fluorescent. Now, these are essentially non-fluorescent until bound to DNA, so you add it as the last step. There's a whole bunch of impermeant nucleic acid dyes, propylium iodide, 7-AD, DAPI. These are, you know, in everyone's toolbox. But if you want some alternative colors, there's some under the Cytox name. And I'll just let you know the SY of that name is a tribute to the organic chemist who developed these, Stephen Yu. So that's kind of a little inside story there that we don't often tell, but it's a great story, and a variety of different laser excitation and emissions that you can use in your panel design. So this is an example of looking at a an image. You just very simple, adding propydium iodide in there, and you can see the dead cells are fluorescent. So you can do this as a very quick check by imaging as well. The second class, these are amine reactive dyes, and they also work on the theory of membrane integrity, but it's a little bit different. So these are protein reactive. The dye will react with the amines on the outside of a live cell. This will result in very dim fluorescence. When the membrane is compromised, the dye will be allowed inside. It will bind the amines on the inside, and you'll have at least a 50-fold increase of fluorescence intensity. Now, what's great about this is that this is a covalent bond, so you can fix this and go on and do, or fix and permit even, and go on and do intracellular work. So just a little bit of a comparison here of why you might choose one or the other. So this is looking at a mixture of live and dead cells with propydium iodide. And so before fixation, you have this nice discrimination of your dead and live cells, but after fixation, eventually the dye comes off. And you have to understand that DNA binding dyes are not covalent. It's a fairly weak interaction, so it is affected by the concentration in the buffer. So that's where these live dead fixable stains are really great, because before and after fixation, you should be getting the same answer. And there's a lot of different colors. So I showed a lot of options for the DNA impermeant nucleic acid dyes, and there's a bunch of different options here as well for the live dead fixables. So some of the tips and tricks here. With the impermeant DNA dyes, add it as a final step and don't wash it out. DNA binding dyes have a very broad inherent emission, so it's a really good idea to look at a spectroviewer to see what's the best detector and where you might run into problems if you have one of these in your panels. And there really is not a good compensation control, but I just like to use dead cells or fixed cells labeled with the dye that I'm using. The amine reactive dyes, because they're amine reactive, remember not to have protein in your buffer while you're labeling with the dye. After you label with the dye, you can wash and do other buffers that have protein in them. Remember that the live cells will have a dim fluorescence that's above the autofluorescence of the cells. These have been validated with aldehyde fixatives. It doesn't mean that you can't use another type of fixative, but I would make sure and test your system out to make sure that you're getting the same results before and after fixation. You don't have to fix your cells to use these dyes, and they do have a more narrow emission, so you have a better panel design with these. And there is a compensation bead, the ARC beads, amine reactive compensation, and these can be used along with your antibody capture beads to set your compensation. So there was a little bit of talk about ROS this week, but I didn't see any of these dyes being used, so I thought I would talk about these. So we have some newer ROS reagents under the name CellRox, and these are fluorogenic probes. It's oxidized from the fluorescent to a fluorescent form in the presence of ROS. It can be labeled in buffer or complete media, and you don't have to have a serum-free media for these. There are 3 different kits or 3 different colors of the CellRox reagent. But they are not the same molecule with just little tweaks for fluorescence change. They are actually different molecules and have different types of reactivity. So here the CellRox green. This is basically a DNA binding dye. Then we have Cellrox Orange and Cellrox Deep Red that are in the cytoplasm. So that's important to know in your design. And if you're really into ROS detection, you may want to study this chart a little bit and see which one of these is the most appropriate for your particular application, because they do have a little bit different reactivity. So the Cellrox Green, of course, this is using the FITC detection channel, and here's a plot or an image of Cellrox Green with Herxt with a number of different controls. The flow cytometry kit also includes some controls, so you can treat your cells with tert butyl hydroperoxide, and there's anyway, you can treat your cells with a positive control and a negative control, and then you can set your voltages appropriately for your particular cells, and your cells of interest will hopefully fall in between there. Cellrox Deep Red, this is actually my favorite. This is in the APC detection channel. Because it's not a DNA binding dye, it's actually fixable with or it's compatible with fixation. Here we have an image of the Cellrox Deep Red is this purple area here with a Cellmax Orange and a Cyto Green nuclear stain. And then finally, Cellrox Orange. So this is compatible with some of the newer green and yellow lasers. And again, this is compatible with oh no, it's not compatible with fixation, but it is a cytoplasmic readout. Any questions on Cellrox? All right. So apoptosis, don't worry, I'm not going to talk about all of these. But there are a number of different reagents that can be used for the detection of apoptosis. And, there's kind of the earlier mitochondrial probes and the middle membrane permeability, translocation, caspase, and then finally at the end here, there's the membrane integrity. Each one of your particular systems will have its own course. I am not going to talk about JC1, but I do want to, this is a great ratiometric dye, but I do want to call out a publication from the Costa Rica lab with Sarah, where it's from current protocols in cytometry using a JC1 with a 2-laser excitation, so you don't have to worry about compensation. So it's a really good way to resurrect an old dye. Huh? I know. Oh, older than me. Oh, I didn't know that. Oh, that's good to know. So I'm just going to talk about a couple of these. You can also very easily look at the morphology of cells, especially cultured cells, and just by morphology, the control cells, they look very happy. They're the same size and the same shape, and when they've been treated to undergo apoptosis, they look a little ragged and have a lot of variability of size and shape, and eventually you'll get this sort of blebbing phenomena. So some older dyes here that have been around in the molecular probes catalog for many years. They've been used in imaging for a long time, but more recently they've been used in flow cytometry because of some of the newer lasers like the 561. So this is tetramethylrhodamine methyl ester and ethyl ester. Both of those are very similar. This is a really exquisite dye for mitochondrial health. So you get a very bright fluorescence when it's a healthy mitochondrion, and as the health degrades, the fluorescence goes away. Another kind of old-time probe is mitoprobe DIC15. What I like about this is it's in the APC detection channel. And again, you have healthy mitochondria that have a nice fluorescence intensity that is lost with the loss of the mitochondrial health. Some of our newer caspase reagents are under the name Cellivant, and we have two different colors of the Cellivant caspase 3,7. So this is actually a nucleic acid dye, and it is conjugated to a DEVD peptide. And it's cell permeant, so it gets into the cell. If there is an active caspase present, it will cleave off the DVED peptide. It will free up the nucleic acid dye, where it will go to the nucleus, bind the DNA, become very brightly fluorescent. This is a really easy probe to use. You don't have to wash it. If you're combining it with some other probe, you can do some washing, and you can add it into complete media or buffer. So, I mean, you can look at this image and know that these cells are, by morphology, looking pretty good, and this little guy here is definitely suffering, and that's where the green fluorescence here is, and of course with flow cytometry you get a nice positive region there. You can also do this very easily with some of the very basic flow imaging systems, and here you have a bright field with our control cells, and you can see some background apoptosis combined with a live nuclear dye. You can look at all of the cells that way, and then with induction of apoptosis, you can of course see all of the additional green there. And with flow, of course, the ability to multiplex is a new thing for a lot of these functional assays, but I would recommend you adding in some sort of dead cell stain. So here we get our population of live, apoptotic, and necrotic cells. You can combine things like cell rocks for ROS detection with caspase, and this is an example untreated. Here we have the Herxt as a nuclear label, and with staurosporin treatment, at 2 hours we see the cell rocks reagent come up, and then after 4 hours we're starting to get expression of the caspase. So this is a movie that's showing the mitochondrial health probe, and it's very healthy, and it goes away as the caspase comes up. Yeah, okay. Maybe. There we go. So the caspase red, this is a newer reagent that was just released in the last couple of weeks. So this is using the APC detector, but it has the same mechanism as the cell rocks green. And this is an example of compatibility of the caspase red with GFP. So this is expressing a gentamicin-resistant GFP and then treated with gentamicin. And you can see that the cells that are not expressing the gentamicin-resistant GFP are undergoing apoptosis with the treatment. So here's another movie showing tetrabethylrhodamine methyl ester with Cellivant. So the mitochondria are very healthy and happy and bright red, and with the induction of apoptosis, you can see that the red fluorescence goes away and the green fluorescence of the apoptotic marker comes on. So this is in flow cytometry, we look at time points of this. I'm going to play this again. And this time I want you to look for the green cells that kind of float off. And as the apoptosis continues, you see some of these green cells just float off. And this is very typical with cells that are on a surface. As they die, they float off, they detach. So one of the things, if you're doing flow cytometry, is you might want to collect the supernatants so you really have all of the cells that you started with and get a full picture of apoptosis. So I talked about the live dead amine reactive stains. So here it's actually a part of the assay. So this is using an antibody against cleaved caspase 3. And so then we're looking, of course, that's an intracellular marker. So labeling with one of the amine reactive dyes first. And you can then look at viable cells and early apoptotic and late apoptotic cells. So I did mention that antibodies can nonspecifically bind dead cells, and this is a dead cell assay. So just be a little cautious when you're adding in antibodies to any kind of system where you're looking at dead cells, and the earlier in the apoptotic pathway is probably okay, but at some point you're going to start binding dead cells nonspecifically. So this is kind of an unknown or not used very much ratiometric dye, the violet ratiometric membrane asymmetry probe. The probe itself is F2N12S, and that stands for this chemical name. I'm not going to try to say it. So this is one of those dyes that, membrane have this double layer of lipids with most of the phosphatidylserine and phosphatidylethanolamine on the inner leaflet, and with apoptosis this flips to the outer leaflet. The phosphatidylserine flipping is often detected with annexins. But what happens with this flipping is the outer leaflet also undergoes a change of charge. And this particular probe intercalates into the outer leaflet of the membrane, and it detects that charge, or the change in the charge. And it is ratiometric, so it is violet laser excited, and healthy cells have an orange fluorescence. And as they undergo apoptosis and die, the fluorescence emission changes to a green fluorescence. So this is orange versus green here. This is a pretty easy probe to use. You add it to your live cells and incubate for 5 minutes, and then you can start acquiring on your system. Ratiometric dyes are, there's not too many of them these days, but they're really great to use because they're somewhat self-calibrating. They're very forgiving of concentration and instrument variation and mass of the cell. And unlike annexins, there is no special buffer required for these. With annexin, you do need to have a fairly high level of calcium in your buffer. So most instruments these days have the ability to do derived parameters, and so you can do a ratio of the fluorescence change and put everything all on one plot. So here's our control cells, our apoptotic cells. You can see the shift here in the violet laser, orange to green. And if you do a ratio, you can do it either orange over green or green over orange. And in this particular case, we have our live cells here, apoptotic, and then combined with a dead cell stain, you can identify those very late dead cells. So, with all this multicolor flow cytometry and 18 colors and 28 colors, I'm we're still kind of in the baby steps of combining a lot of these different functional assays. One of the challenges with this is the sample prep, trying to work out different conditions and different temperatures and different buffers that each of those might require. But there's a lot of choices now for apoptosis, and really a multi-parametric approach gives you a lot more information than just a single assay. So the video I showed earlier, so in flow, we're just looking at different time points. We don't have the ability to measure a time lapse like you do in imaging. So we're actually looking at different time points of this. And as you're developing, especially apoptosis, I'd really suggest doing some sort of a time course to really understand the model you're using and the particular assays that you might want to use for apoptosis and the times that those come up in. You can certainly do this with a reverse time course, so you can harvest and stain the cells all at the same time. So here is a cell event caspase versus annexin versus a dead cell stain, and caspase versus a dead cell stain. It's kind of hard to see the populations there, but this is a fairly easy combination to do, and certainly something that I'm sure everyone in this room could do. So this is using diIC15 with a caspase 3 and a dead cell stain, and you can start looking at untreated versus treated, and look at the changes in mitochondrial membrane potential. Here we see that they precede the membrane permeability and even caspase activation. All right, so we're going to shift gears a little bit and talk about some applications of click chemistry. So what is click chemistry? This had its origins when folks were looking to build chemical libraries of very small molecules. And this name stuck to one particular reaction, and this is a copper-catalyzed azide alkyne cycloaddition. And so this is where you have these two small molecules, an azide and an alkyne. And in the presence of copper, it forms this nice structure with a covalent bond. Now, this reaction is highly efficient. It's rapid, and that's kind of the name. It happens like a click. It's stable in physiologic conditions, and most importantly, bioorthogonal. So these reaction components are not normally found in nature, so they are very selective in their reaction. So it's also a very versatile labeling moiety, where you can have the detection probe in either an azide or an alkyne, and depending on what it is that you're measuring, you'll have the other component for the detection. And I'm going to be describing one application from each of these. And the first is a proliferation assay using the Clickit EDU. And of course, this is a DNA content histogram. This is, I think, Herxt. And with a software deconvolution, you can really see that there's these overlapping phases with the S phase in the middle. But sometimes you have plots that look like this and may be a little difficult to determine what the S phase is. These are actually cells that have been treated with blockers, which is a very common treatment. It's interesting in looking at these, they're very different plots, and they model quite differently, but the S phase on these is about the same. So it's, really wonder, is that accurate or not? So these are thymidine analogs. Procedures for these have been around for many years. First of all, with tritiated thymidine, and in the 1970s, the BRDU method was developed. And what I'm going to be talking about is EDU, or ethynyl deoxyuridine. You can look at the structures here, and they're all very similar. In fact, it's just this terminal end here is where the difference is. So this concept can be applied to the labeling and detection of DNA with click chemistry because the alkyne, let's see, is incorporated into the DNA, and then with a click reaction, incorporating a dilabeled azide in the presence of copper, then you get this nice covalent labeling of your DNA with a fluorescent probe. And to really understand what an advancement this technology is, I'm going to go through some graphics here. So I don't think I need to mention, tritiated thymidine and radioactivity. I hope no one here has to do that. BRDU is definitely an advancement over that. So it's incorporated into actively synthesizing DNA. And it's detected with an antibody. However, the antibody is fairly large, and it cannot access the incorporated BRDU. And so because of that, the DNA needs to be denatured. And there's been a lot of protocols developed over the years to denature the DNA. It's the more tricky step of the assay. But once it's been denatured, then the antibody can come in and bind to the incorporated bromodeoxyuridine. So EDU, also a thymidine analog, incorporated into actively synthesizing DNA. However, it's detected with this very small molecule, dilabeled azide, and it does not require any sort of DNA denaturation. So why do we care about this? Well, it makes a very simplified protocol. So you can incorporate the EDU and then just follow fix and perm procedures. So once you fix and perm, then you apply the click reaction in your labeling mixture, and you're done. And there's very little optimization that is required for this. I do call out aldehyde fixation and detergent perm because that is what is in the kit that is offered. But it is compatible with absolutely every fix and perm procedure that I've seen used with it. So I'm pretty confident that it has no problems there. And then you get plots that look like this, where the EDU, if you put a region here, these are the cells that are in S phase. Combined with a DNA binding dye, you get this very typical pattern where these are the cells, G0, G1, G2M, and then these are all the cells in the S phase. So here's an example before Cyto last year in Glasgow. We had a workshop, and we ran this assay. And this is the type of plot. I'm actually going to go to this next slide because it shows the type of gating. So looking at the main population of cells, we added in a live dead dye first to exclude the dead cells and gate on the live cells, and then looking at singlets. And then from that, we're looking at both the DNA content and the EDU signal, and then putting those all together to look at all the populations. So the EDU Click-It kit has been out for a few years, and there's a new version of it. And the reason why is that the original kit did have some limitations. It did have a detrimental effect on the fluorescence of PE tandems, and quantum dot fluorescence. So these kits are still available, but I would highly suggest that you move over to the Click-It Plus. So this has a new formulation. It's actually the the copper in the click reaction that causes the problems here. But the new formulation takes care of all of that except for it's not compatible with Q-dot fluorescence. And this is now compatible with fluorescent proteins. There was variability of the original kit with compatibility with fluorescent proteins. So the good news is there's really no price difference between these kits, so there really is no other reason to not move to the Click-It Plus. So here's some data from one of my collaborators, Bill Telford, and here he's showing the cells that have been labeled with PE size 7 or PE, and with the original kit, there is no fluorescence detected, but with the Click-It Plus, you can see a nice fluorescence intensity. And this is an additional control where we have the Click-It Plus, but without the copper present to show that we have equivalent fluorescence. And then he's also done some additional work with some common fluorescent proteins. And so here we have live cells with 3 different fluorescent proteins. We're adding in a fix, and we still see fluorescence intensity. When we add in the perm, there might be some decline of the fluorescence intensity. With the classic, the original Click-It kit, you see this sort of variable reaction, but with the Click-It Plus, you get a nice discrimination, and then once again with the Click-It Plus without the copper in, so you can see this is a very nice control. So this is compatible with any fluorescent protein that would survive fix and perm. And there's a number of different colors to match your instrumentation and whatever panel you might want to put this in. So there are a couple of workflow variations that I like to talk about. So first of all, you can add in a live dead fixable stain. Then you can do surface immunophenotyping, fix, and then I like to combine the click and the perm together to save an additional 30 minutes of my time. I call it click and perm, and then go on to intracellular labeling, and then finally do a DNA content dye. It's also compatible with imaging. So here we have an Alexaflor 88 with Herxt as a nuclear dye, and this is HeLa cells. So you can see these are the cells that are actively synthesizing DNA, and these are not. So another application of click chemistry is the cyclic antibody labeling. So traditional labeling mechanisms have this sort of random labeling of amino acids. This can lead to disruption of the antibody binding sites and may cause poor reproducibility, and certainly there is a bit of optimization required. So with amine labels, this is a stable covalent bond to the lysines. The degree of labeling can vary between antibody and fluorophore, and you can have labeling of the binding site. Thiol labelings are another type. These are a covalent bond to cysteine residues, and again, there's variability here. So what we've done is developed a method using click chemistry for a site-specific labeling of the antibody. And this is looking to label the carbohydrate domains here in the FC region of the antibody, which is far away from the antibody binding sites. So I'll describe what this is, and of course this is the antibody, and these are the carbohydrate groups that are coming off of that. So the first thing is beta-galactosidase removes the terminal galactose in the region there. And then GalNAc residues are replacing with an azide containing sugar. And, azide is one of the components of the click reaction. And so now this antibody contains the azide. And we come in then with a dibo label. So the dibo is the alkyne. And then in a copper-free click reaction, these join in a covalent bond. So this dibo can be conjugated to a fluorophore, a Q dot, a bead, a drug, whatever you want to label it to. And now your antibody is labeled very specifically in the FC region of the antibody. We do have kits for this, so you can label antibodies yourself, and this has a very consistent degree of labeling of 3.5, so there really is not a lot of optimization that is required. With traditional labeling kits, there is a fair amount, so that's one of the biggest advantages. So this is looking at just a CD4 RPE. This is a traditional off-the-shelf product that's gone through all kinds of optimizations and QC, and this is a single attempt at a side-click conjugation. So I thought that was enough for today. Anyway, thank you so much.
We have time for a lot of questions.
Or none, because I know everyone.
We have 1, 2, 3. What about JC9?
I haven't used JC9 very much, but I know people that like to use JC9 and even JC10. Do you? None of them are new. Have you used any of those? Yeah.
It's one question on the side click click chemistry to bind to couple fluorochromes to antibody, which I think it's great. Is it working well on all kind of antibody raised in different species like rat, hamster, mouse? Or is it something specific?
There are some of the antibodies that I don't think it works on. I don't think like IgM antibody, but mouse or human or
all IgGs would work.
I believe so.
Okay. And then can you control the number of P then by that way that are grafted onto the antibody? So can you really make sure on the number of fluorochromes that are bound to the antibody?
Yeah, so there's only 4 possibilities.
Yeah.
So, if it binds all 4, it's, you have a degree of labeling of 4, and if it binds...
Is there a way to control that you have only 1 or 2?
I think we're pretty sure that it's a consistent 3.5 in that range, so I can find out, though. I can get back to you.
No one else? Okay, thank you.